Hello and welcome to Sam Biology class. Friends, transcription is a biological process by which the enzyme RNA polymerase synthesizes RNA on a DNA template. To transcribe a gene, RNA polymerase proceeds through a series of well-defined steps which are divided into three phases. First, initiation. Second, elongation. And third, termination. Initiation begins with the binding of RNA polymerase to a specific promoter. A promoter is a specific DNA sequence which is usually located upstream from the transcription start site. Here +1 represents the transcription start site from which the process begins. The binding of RNA polymerase with the promoter result in the formation of a closed promoter polymerase complex. This closed complex then undergoes structural changes which is required for initiation to proceed. As in case of DNA replication, DNA around the transcription start site unwinds. That means there is localized strand separation. So base pairs are disrupted. As a result, an open promoter polymerase complex is formed. The localized melting of DNA around the transcription start site produces the transcription bubble. This bubble is a molecular structure where the two strands are separated. Its size ranges from 12 to 14 base pairs. Again, like replication, transcription also proceeds in 5' to 3' direction. That means ribonucleotides are added to the 3' end of the growing RNA chain. Again, like replication, transcription also proceeds in 5' to 3' direction. That means successive ribonucleotides are added to the 3' end of the growing RNA chain. However, unlike replication, in transcription, only one DNA strand is used as a template. The strand is called as template strand or non-coding strand, and the other DNA strand is called coding strand or non-template strand. The coding strand is identical to the synthesized RNA chain except that in place of uracil in DNA, thymine is present. The choice of promoter will determine as which stretch of DNA will be transcribed. This is the first step where regulation is imposed on the process of transcription. Second phase is elongation. After synthesizing a short RNA chain of about 10 ribonucleotides, RNA polymerase shift into elongation. This transition requires further conformational changes in RNA polymerase which enable the enzyme to grip the DNA more tightly. During elongation, RNA polymerase perform an impressive range of tasks in addition to RNA synthesis. Number 1, RNA polymerase catalyzes localized unwinding of DNA in front and reannealing of DNA behind. Number 2, it dissociates the growing RNA chain from the template as it moves along. And number 3, it also performs proofreading. By contrast, in the process of DNA replication, a number of enzymes are required to perform similar range of tasks. Last phase is termination. After completely transcribing the stretch of DNA, RNA polymerase stop transcribing and release the transcript from the template. In some cells, there are specific well-characterized sequences that trigger termination, whereas in other cases, it is less clear that what instructs RNA polymerase to stop transcribing and get released from the template. Friends, this is just an overview of transcription. Further, we shall discuss about the greater detail of this process.